And our first speaker today is Sonia Jakowlew. She got her Ph.D. at Rutgers University, then she did a postdoctoral fellowship in France. Subsequently, she came to NCI in the 80s, and she started working with Michael Sporn and Anita Roberts on TGF Beta. And she's going to talk to us about that today. Currently, she's in the cancer training branch, and the title of her talk is Transforming Growth Factor Beta and Lung Tumorigenesis. Sonia.
Okay, thank you, Harry. Well, it's good to be here, and um, today I'll be talking to you about transforming growth factor beta and lung tumorigenesis. And I will just touch briefly on the lung cancer disease and let the heavy lifting for lung cancer disease to Dr. Yves, who I believe will be speaking in a few weeks to the class. So, like most past years, lung cancer was determined to be the most common cause of cancer deaths among both men and women in this country last year. There were over 221,000 new cases of lung cancer that were diagnosed in this country last year among both men and women. And just slightly over 158,000 deaths due to this disease. So it's a serious, significant disease. Because lung cancer is primarily caused by tobacco smoking, and the incidence of smoking has decreased in this country, thankfully. Most cases of lung cancer now occur in former ex-smokers. However, there are many, many patients who have lung cancer who have never smoked. So there are other perpetrators of lung cancer, which include exposure to radon, exposure to coal and coal dust, and environmental conditions that precipitate inflammation in the lung. So the 5-year survival rate for lung cancer is still a dismal less than 15%. So a lot of work has yet to be done on this disease. Now, I started on transforming growth factor beta, or TGF beta for short. My interest began in the early 80s. And TGF beta has been shown to be what's called a multifunctional regulator of cell growth. It's been shown to be a rather potent inhibitor of the growth of most normal epithelial cells and shows widespread tissue expression. TGF beta has been shown to play a pivotal role in maintaining epithelial homeostasis and has been shown to be associated with various types of cancer, including lung cancer. So, and finally, TGF beta is been shown to have what we call display context-dependent inhibition or stimulation of cell proliferation and neoplastic transformation, depending on the environmental conditions. So TGF beta is an attractive candidate for new therapeutic intervention approaches into cancer. Now, to understand TGF beta, you have to go back to the beginning. And TGF beta has its roots in another growth factor called sarcoma growth factor. The sarcoma growth factor was identified in the early 80s as being a polypeptide that was secreted by Maloney marine sarcomavirus transformed mouse fibroblasts that stimulated normal rat fibroblasts to form colonies in soft agar. So this transformation assay was used as the assay for this growth factor by Joe DeLarco and George Todaro at the NCI in the 70s. And shortly after sarcoma growth factor was identified, it was determined that there were actually two different proteins that made up this growth factor, and they were called transforming growth factors. They were isolated from the Maloney sarcoma virus transformed cells that Joe DeLarco and George Todaro used to identify sarcoma growth factor. And one class was shown to compete with epidermal growth factor, EGF, for receptor binding. And this class was called TGF alpha. Another class was shown to not be able to compete for EGF binding, but could form colonies when in the presence of EGF. And this protein was called TGF beta. So sarcoma growth factor is really is a growth factor that is really composed of two different proteins that fortunately or unfortunately share the name of TGF. They're very different proteins. They do very different things. So this was reported by Anita Roberts and Michael Sporn in the early 80s. So following this, the Sporn lab hastened to purify TGF beta from a variety of different sources, including human platelets, human placenta, as well as bovine kidney. Now, to demonstrate a purification from bovine kidney, batches of 100 grams of bovine kidney were typically obtained from the slaughterhouse and extracted for several hours with acid ethanol, centrifuged to get rid of the waste, and then was precipitated overnight with about 50 liters of ether ethanol in the cold room. Next morning, the precipitate was then dissolved in 1 molar acetic acid and then applied to 80 liters biogel P60 columns. 1 liter fractions were then collected, and you know what a 1 liter is. And these were lyophilized and redissolved for further chromatography purification. So typically from 100 grams of tissue, they would get 6 micrograms of TGF beta. Okay? So this slide shows you to get an idea of the columns, the enormity of the columns that were needed to purify TGF beta. And on the right, lower right, if I can get this to work, okay, this is a 55-gallon barrel to give you some idea of the size. Now, the assay for TGF beta included the growth of normal rat kidney cells, NRK cells, and soft agar. So typically, to perform this assay, an agar plate was made, and then a mixture of media, serum, NRK cells, EGF, and sample, which may or may not contain TGF beta, was applied to the agar, then allowed to incubate at 37 degrees for a week, then stained, and the colonies were then counted on the plates using an Omnicon image analysis system, which is shown at the bottom of the slide here. That was state-of-the-art at the time. So if no TGF beta is present in the sample, then one would see no colonies, and no colonies would be counted. If TGF beta is present in the sample, though, colonies would appear over the 1 week, and if they were of a specific size or larger, they would be counted. So after the final high-pressure liquid chromatography purification, samples were then put on a polyacrylamide gels and ran. And purified TGF beta migrated as a 25,000 molecular weight protein on single band on the gel. So Doctors Michael Sporn and Anita Roberts are credited with the discovery and purification of TGF beta at the NCI. And I had the good fortune to work with both of these investigators for 6 years, and it was one of the happiest, most informative, exciting times of my life, of my research life. And unfortunately, we lost Anita Roberts in 2007 from gastric cancer. When she passed on, she had the enormity of being the second most highly published female investigator in the world. And Michael Sporn has since left the NIH, and he's a endowed professor of medicine at the Dartmouth College in New Hampshire. And he's at the ripe young age of 84 and still going and studying TGF beta and triterpenoids. So let's hear it for the youngsters. Okay? So following the identification and purification of TGF beta, the sequence of TGF beta was determined, and actually it was found that TGF beta is initially formed as what is called a pre-pro molecule of about 391 amino acids. It contains a signal peptide sequence at the N-terminus, a middle portion which has been called the latency-associated peptide, or LAP for short, and a mature TGF beta sequence at the C-terminus of 112 amino acids. Now, the amino acid sequence has been determined, and I give you a pretty colored picture denoting the 9 characteristic cysteines that highlight this class of proteins. And I'll talk about that a little later. So it contains a signal peptide, therefore this is a secreted protein. Now, the crystal structure of TGF beta was determined in the early 90s. This happens to be a representation of TGF beta 2 because for unknown reasons, TGF beta 2 is much easier to crystallize than TGF beta 1. So TGF beta 2 and TGF beta 1 occurs as a dimer of 2 identical monomeric chains held together by an interchain disulfide bond with a hydrophobic pocket that other proteins can interact with. Now, TGF beta is a canonical member of what is called the TGF beta superfamily. This superfamily contains, in addition to the TGF beta isoforms, the BMPs, GDF, growth differentiation factors, as well as activins and inhibins. Now, to kind of summarize what I've been talking about, the TGF is a 25,000 molecular weight disulfide bonded homodimer. There are 3 highly homologous isoforms that have been identified in humans, and these are called TGF beta 1, 2, and 3. They're separate proteins, distinct proteins, but they are homologous. In addition, TGF beta 4 has been identified in birds, and TGF beta 5 has been identified in amphibians, and TGF beta 4 and 5 have most closely homology to TGF beta 1. The principal sources of these TGF beta in the human are platelets, bone, and spleen. Most cells have been shown to express TGF beta as well as its receptors, and TGF beta is usually secreted in a latent inactive form, which must be activated before TGF beta can act on expression or do any of its activities. And the superfamilies of TGF beta encompasses not only the TGF betas, but activins, inhibitors, BMPs, and GDFs as well. Now, members of the TGF superfamily control central control modules for a number of biological processes, including development, immune system, reproduction, angiogenesis, aging, tissue repair, metabolic regulation, and homeostasis. So it spans a wide range of activities. The major biological responses regulated by TGF beta, they're very numerous. TGF beta can inhibit proliferation of cells. It can regulate apoptosis, differentiation, immune cell function. It can stimulate accumulation of extracellular matrix, and it can promote chemotaxis of certain cells. So is there anything else that TGF beta can't do? Okay, that was the question in the early 80s. So model for TGF beta pathway has been developed over the years, and this we call the SMAD-dependent pathway because of the involvement of the SMAD proteins in this pathway. So this begins by the TGF beta ligand shown in blue at the top, and this can be TGF beta itself as well as activin inhibins, BMPs, etc. Binds first to a receptor called the type, lost my mouse. Oh, sorry. Okay. The TGF beta ligand binds first to the type 2 receptor, which is constitutively phosphorylated. Once this binds and okay. Once this binds, it then forms a complex and recruits another molecule called the TGF beta type 1 receptor to form a ternary complex. The type 2 TGF beta receptor then transphosphorylates type 1 receptor, and that enables it to then be transduced to other proteins called the SMAD proteins. Now, depending on what the ligand is, the SMADs are specific to the ligands. For TGF beta and activin, the SMADs are SMAD2 and SMAD3. For the BMPs, we have SMAD1, SMAD5, and SMAD8. So these carry the signal, are transphosphorylated, and then deliver the signal to another SMAD called SMAD4, which is called a common SMAD. So this SMAD is then able to transduce the signal into the nucleus, where it can then participate and affect transcription. So the whole process can be circumvented by what are called inhibitory SMADs, SMAD6 and SMAD7, which circumvent the whole process. Okay, so there are regulatory SMAD that make the TGF beta pathway go forward, and there are inhibitory SMADs that stop the pathway itself. So in addition to the laboratory, clinically, TGF beta has been studied in the clinic as well. It's been shown that TGF beta is a type of tumor suppressor. For example, germline mutations have been shown to occur in various components of the TGF beta pathway to cause familial predisposition to cancer. And this has been shown quite nicely with SMAD4 in juvenile polyposis syndrome. The second example is the TGF beta pathway components may be somatically mutated or deleted in some human cancers. Now, the type 2 TGF beta receptor has been shown to be mutated in human non-polyposis colorectal cancer, while SMAD4 has been shown to be deleted in many cases of pancreatic cancer. thirdly, reduced expression of TGF-beta signaling pathway components or overexpression of these inhibitors have been shown to be associated with cancer disease progression. And for this, it's been demonstrated that the type 1 and type 2 TGF-beta receptors can be overexpressed, while SMAD7 can be, as an inhibitor, can also be overexpressed in several types of cancers. Now, in addition to being a tumor suppressor, clinically, TGF-beta has been also shown to be a possible tumor promoter in the clinic. So elevated levels of TGF-beta1 has been shown to occur in many advanced types of human tumors and to correlate with metastasis and often with poor progression. And I highlighted a number of tissues in which we see elevated levels of TGF-beta1 in tumors, including the lung. And shown on the right is a prostatic adenocarcinoma that's been histologically stained for TGF-beta1 with a very specific antibody. And TGF-beta seems to sit at the interface between the tumor parenchyma and the microenvironment, okay? So what's going on? What's the role of TGF-beta in carcinogenesis? Is it a hero, like we all thought in the beginning, or has it turned into a part-time villain? Well, the answer is probably it's both. TGF-beta is a proximal effector of the malignant phenotype. It's also a potent growth inhibitor and tumor suppressor, especially early on in the process. And it's also a pro-metastatic factor. Now, how does this occur? Well, over the years, a unifying hypothesis has been developed whereby TGF-beta can switch from being a tumor suppressor to a pro-oncogenic factor as cancer progresses. So when we start in the normal epithelium, okay, we have a normal epithelium, normal cells. The tumor suppressor activities of TGF-beta dominate over any pro-oncogenic activities. But as changes occur in the genetic and epigenetic context, the level of TGF-beta responsiveness goes down, and as tumorigenesis or carcinogenesis progresses, the expression and/or activation of TGF-beta increases. So when we have a invasive metastatic cancer, then the pro-oncogenic activities of TGF-beta predominate over the tumor suppressor activities. So we have essentially a tumor suppressor switching and turning into a pro-oncogenic molecule. Now, in addition to the SMAD pathway, there are a number of what's called TGF-beta SMAD independent pathways that enter into the picture. And this includes a number of pathways such as the TAC junk pathway or MAP kinase pathway, the RAS MAP kinase pathway, the RO-ROCK pathway, and the PP2AS6K pathway. Now, we were particularly interested in the RAS MAP kinase pathway because the KRAS proto-oncogene is very important in lung cancer. The KRAS gene shows an activational mutation in at least 25 to 50% of human lung tumors. It's been shown that mutation of even one allele of KRAS can increase the appearance of lung lesions, and this can occur early on. And it's been demonstrated that there's actually a crosstalk that occurs between the SMAD-dependent pathway and the RAS signaling pathway in several types of cell types. And activation of the RAS pathway can modulate TGF-beta signaling through the SMAD. And lastly, in vitro studies in cell lines and several cell lines have shown that the TGF-beta dominates over the mitogenic effects of RAS. But when RAS gets activated, this can override the anti-proliferative effect of TGF-beta and turn it more into a pro-oncogenic factor. And this is a problem. So this slide represents at least 4 different ways that the tumor suppressor can turn into a tumor promoter. How this occurs? Well, we have the normal condition where where in normal cells where TGF-beta interacts with the type 1 and type 2 receptor through the SMAD pathway and other pathways, and to result in tumor suppression. But if something occurs and say, for instance, the decrease in the amount or level or expression of the type 2 receptor, or there may be hyperactivation of the SMAD, of the MAP kinase pathway with activated RAS, or there might be decreased SMAD levels or activity, or there may be other things that may be happening in the tumor suppressor arm of this pathway. Then you have a tumor promoter situation in lieu of a tumor suppressor situation. So we have been kind of interested in the hyperactivated MAP kinase pathway in our studies. So the broad goal of my laboratory was to determine the role of TGF-beta in the development and malignant transformation of lung epithelial cells. And this work was done in the epithelial carcinogenesis section of NCI. So we had 3 objectives. The first, to examine the effect of TGF-beta 1 deletion and KRAS mutation alone and in combination on lung tumor incidence and pathology. Then to determine the early events that might occur in the development of lung lesions and how they progressed. And then to identify any potential signal transduction pathway changes that may occur with increasing levels of tumorigenesis. So to do this work, we employed 4 mouse models: the AJ mouse models, the C57 Black 6 TGF-beta 1 heterozygous mouse, the AJ Black 6 TGF-beta 1 heterozygous mouse that we generated, and the TGF-beta heterozygous KRAS latent activatable mouse that we generated in collaboration with Tyler Jacks of MIT. So we had 2 questions. The first question, does lung tumorigenesis affect the TGF-beta signaling pathway? And the reciprocal question, does the TGF-beta signaling pathway affect lung tumorigenesis? So we chose the AJ mouse model because this mouse is very susceptible to chemically induced lung tumors. The tumors that develop in a time-dependent fashion. They progress from hyperplasia to adenoma and then on to carcinoma, very similarly to what occurs in humans. And the carcinomas that develop are histologically similar to the human lung adenocarcinomas, which are the chief carcinomas that appear in human lung cancer. And unfortunately, the same molecular mutations occur in both human and mouse lung tumors, such as overexpression of KRAS and loss of p53 expression. So we use ethyl carbamate as our lung-specific carcinogen. And ethyl carbamate can go through two pathways. Where there's a detox pathway that results in just harmless ethanol. There's also a bioactivation pathway that proceeds through CYP2E1 in the lung, and this results in vinyl carbamate and vinyl carbamate epoxides, which are then able to bind to macromolecules and cause nasty effects. So, for production of tumors in these mice, we injected ethyl carbamate in the 2-month-old mice, and then sacrificed them at monthly intervals for up to a year, with 20 mice per sacrifice for statistical reasons. Now, shown on this slide is an immunohistochemical staining of tumors at various stages of tumorigenesis, and stained with specific antibodies to TGF Beta 1 and the type 1 and type 2 receptor protein. And you will see in the left hand and right hand middle panels that there's distinct brown staining for TGF Beta 1 and the type 1 receptor throughout these tumors. However, on the right for the type 2 receptor, staining for this protein is well diminished in the 2-month tumors, and by 4 months and 8 months, it's virtually gone. So we see decreased expression of the type 2 receptor protein in tumors with advancing tumorigenesis. This is shown more clearly on this slide if you'll direct your attention to the left side of the screen, specifically panels B and D. The red arrow shows distinct expression of the brown staining for the type 1 and type 2 receptor at virtually identical levels in the tumors. But if you look at the tumors in comparison, there is intense staining for the type 1 receptor, while there's very diminished staining for the type 2 receptor. This is also, we looked at the expression of the mRNAs for these proteins as well, and using 5 cell lines, 5 mouse lung cell lines, we show expression of the type 1 message at various levels in these cell lines, while the expression of the type 2 receptor message is virtually gone in these 2 cell lines. And I highlighted the 5th cell line, PCC4, because these were derived from a ethyl carbamate-induced mouse lung tumor. So this mirrors what we're seeing in the AJ mouse. So we're just showing decreased levels of the protein and mRNA for the type 2 receptor with advancing tumorigenesis in these mice. This is we also looked at lung tumors from benzpyrene-induced lung tumors, and there we see virtually the same thing using both immunohistochemistry and in situ hybridization. If you'll direct your vision to the right side of the screen, we see decreased levels of expression of the protein for the type 2 receptor, decreased expression of the mRNA for the type 2 receptor in comparison to the type 1 receptor and the TGF beta ligand itself. Now, when we stain for these proteins in normal bronchioles, the levels are almost identical in all. So there's decreased expression of the protein and messenger RNA for the type 2 receptor in these tumors. So our mouse model reflects that there's truly, with with decreased expression of the type 2 receptor, we see a good correlation with promotion of lung tumors in our mouse model system. So our next question was, does deletion of TGF beta 1 affect lung tumor genesis? And for this, we shifted to the C57 black 6 TGF beta 1 mouse because the TGF beta 1 knockout mouse was engineered and generated in the early 90s. And it's born, it thrives, but at about 3 weeks of age, it starts to get very sick and succumbs to a general wasting syndrome. So this mouse isn't going to be very good for carcinogenesis studies. But the knockout mouse, the TGF beta 1 heterozygous littermate, is born, thrives, and survives. It doesn't succumb to a wasting syndrome. So in collaboration with Lala G. Wakefield, we did a study. We looked on, because she was interested in liver tumor genesis, we challenged these mice with dimethylnitrosamine, which is specific for liver tumors. And we showed that expectedly there was an increased level of tumor genesis in the liver of the heterozygous mice, shown in red, compared to the wild-type mice. Now, unexpectedly, if you focus your attention on the right side of the film, we saw an even greater number of lung tumors forming in these mice. So these heterozygous mice had an increased tumor incidence of lung tumors in their lungs. So we kind of asked, this is very surprising. So in order to be able to look at in a better mouse model, we crossed AJ mice, which were wild type for TGF beta 1, with C57 black 6 TGF beta 1 heterozygous mice, and to derive two different genotypes, TGF beta 1 heterozygous mice and the wild type littermates in the F generation. Our plan was then to treat them with carcinogen, ethyl carbamate, and look at the lung tumors. So to establish that we were on the right pathway, we isolated lungs. These are lungs from wild type and heterozygous mice, and stained them for the TGF beta 1 antibody, and as well as looked at in situ hybridization for the TGF beta 1 ligand as well. And if you look at the top panel, the middle panel shows decreased expression of the TGF beta 1 protein and message in the heterozygous mice compared to the wild type mice, as was expected. In addition, we performed northern blotting on these mice lungs, as well as competitive RT-PCR, and showed reduced expression of the TGF beta 1 ligand in the heterozygous mice compared to the wild type. So our premise was working. Okay, we had the right mice. So we went along just as we had done with the AJ mice, this time injecting them with ethyl carbamate as before in 2-month-old mice, with groups of TGF beta 1 heterozygous mice and wild type mice, and sacrificed groups of 20 mice over a 12-month period. And when we did this, we extracted we got the lungs and looked at the microscopically for hyperplasia, adenoma, and carcinoma. And in panel A, you'll see that in panel B, you'll see that in the TGF beta 1 heterozygous mice, shown in red, there's a definite, we see the appearance of hyperplasia and adenomas occurring by 1 month, whereas it takes 2 to 4 months before we see adenomas and hyperplasia occurring in the wild-type mice. Now, the striking thing is shown in panel C, where we see carcinomas appearing by 4 months in the heterozygous mice, but it takes almost 1 year, 12 months, before we see any carcinomas starting to appear in the wild-type litter mace. So in these mice, we definitely see an increased tumor incidence and multiplicity, but a decreased tumor latency in the TGF beta 1 heterozygous mice compared to the wild-type mice. This is with loss of one allele of TGF beta. So we then proceeded to stain for the type 2 receptor. And in these mice, unlike the original AJ mice, we see reduced staining for the type 2 receptor protein in the carcinomas, but not in the adenomas or hyperplasia shown in the right panels. Okay, so there's a vast difference between these two mice. Okay, so it seems like the level of the type 2 receptor is maintained in early tumorigenesis, but it's overtaken with increasing tumorigenesis. So we also looked at the relative levels of the type 2 receptor mRNAs with increasing tumorigenesis in these mice, and seems like they're decreasing levels of the type 2 receptor mRNA with increasing lung tumorigenesis as tumorigenesis proceeds from hyperplasia to adenoma and then to carcinoma. So our next question was, does deletion of TGF beta 1 and mutation of KRAS combined affect lung tumorigenesis? And for this, we generated the TGF beta 1 heterozygous KRAS latent activatable mouse. And I won't bore you with the details, but essentially we crossed TGF beta 1 heterozygous mice and C57 black 6 strain because we had to go back to the original because we had a definite problem with the number of offspring generated from just the AJ mice with KRAS latent activatable mice. And this was in strain SV129. And we generated 4 different genotypes. The TGF beta 1 heterozygous KRAS latent activatable mouse, which we call the double mutant, the TGF beta 1 wild type KRAS latent activatable, which we call the KRAS single mutant, the TGF beta 1 heterozygous KRAS wild type, which we call the TGF beta heterozygous single mutant, and the last was the wild type combined. So we let these mice grow, and at 6 months, we started looking at these at 4 months, but these are 6 months long, 6 months excised from 6 months lungs. And in panels A and B, you see these pearly white tumors that are forming on the exterior of the lung. And these are very evident in the double mutant shown in panel A and the KRAS single mutant shown in panel B. Whereas in panel C and panel D, the TGF beta 1 single mutant and the wild type do not really show any tumors on the lung, just an occasional one, very infrequently. We looked at the effect of TGF beta 1 gene deletion and KRAS mutation on mouse survival, and particularly on mortality. And I'd like to direct your focus to plots for A and B. A is this double mutant, and there's significant decrease in mortality, decreased lifespan, and B, which is the KRAS single mutant, is also affected, but less so, compared to the TGF Beta 1 heterozygous single mutant shown in C, and the wild type shown in D. So there's definitely decreased lifespans in the double mutant and the single KRAS mutant mice. We looked at the pathology of these lung lesions, and we showed increased hyperplasia and adenoma in the KRAS single mutant shown in green, but increased development of carcinomas in a double mutant shown in red. Okay, so this is kind of a perplexing here, but it seems that in the single mutant there's kind of a protection up to a certain point, but in the double mutant that protection is gone after a certain time, and you have increased carcinoma development. We stained for TGF Beta 1 and the type 2 receptor proteins in these lung tumors, and shown by the pink arrows, you see decreased expression of TGF Beta 1 and the type 2 receptor in the double mutant tumors. Because the SMADs are intimately involved with TGF Beta, we looked at the expression of SMADs, and I'll focus you on the upper two panels. And in the double mutant shown on the right, we see a definite expedited reduction of the type 2 receptor and definite increase in production of SMAD3, occurring by 2 months compared to 3 and 4 months in the wild type single mutant. So we've performed a number of similar Western blots. In addition to the type 2 receptor SMAD3, we saw reduced expression of the SMAD4 and SMAD7 proteins, as well as increased expedited expression production of KRAS and RAF1. We also looked at the RAF1, which is integral in the KRAS pathway as well. We looked at apoptosis index in the single mutant and double mutant, and shown in red, we see definite reduced apoptosis in the double mutant adenomas compared to the single KRAS mutant. So our mouse models definitely show and seem to correlate that decreased expression and production of the type 2 receptor correlates with increased lung tumor promotion. Activated KRAS MAP kinase seems to correlate nicely with increased lung tumor promotion. Compromised apoptosis correlates with increased lung tumor promotion. And I didn't show it here, but decreased SMAD4 also shows a correlation with increased lung tumor promotion in our mouse models. So I'd like to acknowledge the people that were intimately involved in this study. Jerry Anderson, he's now working at the George Washington Hospital. Yang Kang, he's now with a biotech company in Rockville. Elena Norova and Joseph Pandy and Sarah Umford. And I'd like to thank Tyler Jacks for the KRAS latent activatable mouse that enabled this study. So I'd like to take any questions you might have at this time. Thank you.
So what's the best
Okay.
target?
Wait, okay.
I was just wondering if there were any treatments that have been developed based on TGF beta or if people were sort of working on.
People are working on it. The problem is it's very time consuming to purify, and it's very expensive. Okay. about 10 years ago, there was a great push on to try TGF beta in wound healing. And there were clinical trials that were started to do this, but the problem is getting enough for long-term study, and that's the problem here. And that's the problem with many of these growth factor studies. Okay. So unfortunately, it's still in the pipeline. Yeah.
So what's the best target, the receptor, the ligand, or the SMADs?
Pick your poison. It depends entirely on the situation. In some situations, it's the ligand. And don't remember, although I call it TGF beta all the time, we're talking about the TGF beta superfamily. So there's a lot of involvement of many different molecules here. So what may work in one type of cancer or disease may not work in another type of disease. So it's kind of a lot to kind of suss over in the basket of growth factors. Okay.
That'll do it, Sonia.
Okay, thank you. Okay.
Our next speaker is Yves Pommier. He got his MD and PhD from the University of Paris, 1981. And subsequently, he came to NCI. He's been here over 3 decades. He's an adjunct member of the NCI Developmental Therapeutics Program, so he plays a big role in drug development. He's received the NIH Merit Award for his elucidation of topoisomerase as a target for anti-cancer drugs. He's also an editor on Cancer Research, one of the distinguished journals. He's received the Paul Ehrlich Award. He's published hundreds of manuscripts. He's going to talk to us today about DNA topoisomerases and their poisoning by anti-cancer and antibacterial drugs. Yves.
Thanks, Terry. Okay, so I guess sound is good. So what I did at the beginning of the handout is to point to some references, because this is not a small field, and it's not a field that's going to shrink anytime soon. I think it's in expanding phase. It got a lot of interest in the early years of DNA structure, when it was realized that DNA could be stained with ethidium bromide, and that DNA could be looked under different configurations when it was sedimented in gradients. So some was sedimenting fast, and that was called form 1 plasmid DNA, and some was sedimenting slow, that was called form 2. And then when ethidium bromide came about, and for you is a given, but in those days it was not a given, and agarose gels were developed, it was realized that form 1 was a super twisted DNA, which then was called supercoiled DNA, whereas the form 2 was relaxed, fully relaxed DNA. And therefore, there was a need to understand what the processes were to go from one to the next form. And in the 70s, the first topoisomerase was isolated, first from bacteria, and very soon after from murine tissues. And as of today, I think we have reached the point where I don't think we're going to discover new topoisomerases, although who knows, there are already 6, and it's a handful. So to keep track of this, I've put at the beginning of the presentation some references that are the ones that I will use during the presentation. That's why they are mine, because they refer to the slides. My interest has been, long-standing interest, is to take advantage of the topoisomerases for therapeutic purpose, drugging them. And so this is pretty much updated. And more recently, I took another challenge, which was to write a review, which just came out. So it's not even, there are no pages yet, but I wrote that with John Nittis in University of Chicago. And John is more an expert on topo 2 and on yeast. And so together we wrote that review, which covers more the biology, not so much the drugs really, because this was covered before, but the emerging, new re-emerging role of topoisomerases, eukaryotic topoisomerase in transcription, replication, and now very much in the mind of all of us in genomic stability. So I'll use some of the slides. If you wanted to go deeper into topoisomerase, there's a book in the library which I painfully edited now, but 2-3 years ago. I swear I'm never going to edit another book because it's a lot of work. and I'm not sure how many people read the book. It's lovely to have a book, but nowadays everybody wants something on the fly. But I love books, and this is one nice to have if you want to look at it. There are chapters on all the different aspects of topoisomerases if you like that. So let's get into the topic. So not counting SPO11, which is in germline cell, which is absolutely essential to generate the diversity of who we are, which is a topoisomerase 2-like protein. So not counting this in any of your cells, especially the replicating cell, there are 3 types of topoisomerases and 6 topoisomerase genes in humans. And there are 3 groups. There is the type, so okay, we call them 1, 2, 3, topo 1s, topo 2, topo 3. It stays simple because in each group there are 2 enzymes and 2 genes. Where it gets a little more complicated is the fact that if you look at the types, there are 2 types only of topoisomerases, type 1 and type 2. And types, it defines the fact that the enzyme works by cleaving 1 strand at a time, so that's type 1. And when they cleave 2 strands at a time, it's type 2. So that's not so hard to remember. And then you got an A here, because humans only have type 2A. But if you go to archaebacteria, they have a lot more topoisomerases than we do, have a different kind. So in human, there's only 2A. But you see, human have 1A and 1B. And 1A was named because it was the first type of topoisomerase that was discovered in E. coli. Anyway, so type 1, topo 1. In the topo 1 enzyme, I'll go back later on the details of this. The enzyme this is duplex DNA, so the enzyme, all the enzyme cleave the backbone, and that's a way to relax the DNA or to do whatever it needs to be done to change the structure of DNA. In the topo 1, top 1, there are 2 enzymes, top 1 and top 1MT. 2 different genes, 2 different chromosomes in all vertebrates. In the topo 2, so topo 2 cleave 2 strands, as I mentioned. So instead of having only 1 break on 1 strand, you get a break on both strands. But each strand is cleaved by 1 homodimer. So it's a homodimer in all cases for the topo 2. And there are 2 types of topo 2 alpha and topo 2 beta. We'll come back to that. These proteins are rather big. A single monomer of topo 2 is 170 kilodalton. As a dimer, it's twice that. So it's bigger than a nucleosome. And then the topo 3 cleaves only 1 strand, has 2 genes and 2 enzymes, top 3 alpha and 3 beta. We'll come back at the end because this is the expanding field in the topoisomerase, especially the last one. It not been worked on for many years too much, but it's coming up as being very critical. The transaction for changing DNA structure go through these nicks or breaks in the DNA, which are called cleavage complexes, and the enzyme makes the break, but also ligates back the break. So this is normally very transient. And then there are drugs that poison the breaks. For topo 1 is camptothecin, and topo 2 there are drugs. We'll come back to that. So if you look at the bigger picture now, if you compare humans versus E. coli, so in human you get the type 1As, which is top 3 alpha, top 3 beta. These are their size in kilodalton. The linkage to the DNA, which is covalent, I'll come back to that when they do their transaction. How many times they pass 1 strand around the other, 1 time at a time for the topo type 1A. Type 1B, top 1, top 1MT, about same size. You see the linkage here, the 3' end. This is very different from the 1A and from the 2A. And only passes 1 strand at a time. And then the 2A you get the 2 alpha and the 2 beta linkage 5 prime. E. coli, on the other hand, is simpler in a way. It doesn't have 1b, has only 1a, and it has what is called a topo 1 in E. coli and topo 3 in E. coli. So that's where it gets confusing. It's just historical. So the enzyme. On the type 2 side, on the other hand, E. coli is more sophisticated. It gets 2 enzymes, gyrase and topo 4. Each enzyme is made of 2 genes, and it's a tetramer. So it's slightly more complex, but the mechanics is very much the same. In terms of medicine and the impact on therapeutics, you see among all these enzymes, the topo 1 is an anti-cancer target of drugs that are prescribed every day, the camptothecins, and we'll come back, and new drugs that are upcoming. The top 2s have been used as a target even before we knew that doxorubicin was targeting topo 2 or even a toposide, but they are very, very promiscuous drug in cancer treatment. Doxorubicin is probably the most used anti-cancer drug, and etoposide is very widely used, and mitoxantrone is used not only in cancer, but sometimes in immune disorders. And a big chunk of targeting topo is on the bacterial side. Gyrase and topo 4 are the target of the quinolones, and quinolones are huge. It's a huge impact in biomedicine, including tuberculosis. The resistant tuberculosis is treated with quinolones now. Those target only the bacterial, and they do not touch the host topoisomerase, giving them their selectivity. On the other hand, the drugs that target the host topoisomerases do not target the bacterial topo. So in spite of very similar structures of enzyme, the drugs are very selective. So the biochemical differences between top 1 and top 2. So in top 1, you have top 1, top 1 empty. Top 2, you have top 2 alpha, top 2 beta, gyrase, and topo 4. You could sort of regroup because they have the same mechanics. So the differences is top 1 empty work as monomer. The top 2s work either as dimer for top 2 alpha and beta or tetramers for gyrase and topo 4. When they make their transaction on DNA, the top 1 cleaves 1 strand at a time, links to the 3' end, so it's drawn here, as the topoisomer, and it uses a tyrosine to attack the DNA, makes a covalent linkage, and then there is a nick here in the DNA. This is the OH. And then this can come back. Normally, this goes right back after the DNA has been relaxed. But if you have a drug, that gets poisoned by the camptothecins, the adenoisoquinolines, and the other drugs. On the topo 2 side, so dimer, linkage to the 5' end, 4 base pair stagger. There's always a 4 base pair stagger. It's not like a blunt double-strand break. Linkage to the 5' end. You could see the polarity. 3' hydroxyl. The drugs are specific to topo 2, etoposide, doxorubicin, and for gyrase and topo 4 quinolones. And the difference is that topo 1 is a very economical enzyme. It doesn't burn, doesn't use ATP, doesn't need it. Topo 2 burns ATP and needs it. It requires magnesium. Topo 1 works even on ice, so it's very effective. Topo 2 doesn't work at 0 degrees. And the drugs are specific for each type of enzyme. So there is a good division in terms of the biochemistry and the pharmacology for these 2 types of enzymes. Now, if you compare, so in this table, the 6, and this is the most updated, the 6 human topoisomerases, that will give you their chromosome location, showing you clearly that they're all in different chromosomes, all independent in that way. The name, their size, and now pay attention to this, because our genome is not just our nucleus. Our genome is the mitochondria as well, which is now called chromosome M. And chromosome M, your mommy chromosome, is actually very abundant in a cell. And the topoisomerases deal both with the nucleus. So in the case of topo 1, the top 1, there's a specific nuclear enzyme, top 1, and then a specific mitochondrial enzyme, top 1 MT, For top2A and top2B, they serve both compartments, and they can go to both the nucleus, and they do go both nucleus and mitochondria, and they do their deed on these chromosomes, whether it's in the nucleus or mitochondria. Top3 alpha is both in the nucleus and mitochondria, and top3 beta is in the nucleus and in the cytoplasm. We'll just come back. The drugs, you could see that some of the enzymes are still not being targeted, and we don't have any drugs for the 3s and the 1MT. And then the mechanism you will understand better at the end of the lecture, but the topo1 work by swiveling the DNA, making a rotation, whereas the topo2 make a strand passage with double-stranded DNA, and the topo3s make a strand passage with single strands. And this is the polarity, the linkage of the enzyme, and these are the reactions. Later on, you could go back to this once we have gone through the lecture. So the way the enzymes, what they have in common is the way they break DNA. They all use a tyrosine as the nucleophile to attack the phosphodiester backbone. And depending on the enzyme, it has a different polarity, different geometry. So in the case of topo1, the geometry will link it to the 3' end. And in the case of the topo2 and the topo3, the linkage will be to the 5' end. And it sets the topo1 apart, the top1B, from all other topoisomerases and brings it to the family of the Cre recombinase. And it is somewhat believed that the top1B evolved actually from a recombinase that then was used for relaxing DNA supercoiling. But the most basic topoisomerases are these, the 5' linkage topoisomerases. So let's go into top1 with a little bit more detail. So as I said, there are two top1. There is a nuclear top1, which is the one we most the community is referring to when we say top1. You have to understand this is nuclear top1. And when we discovered top1MT, we made the decision at the time not to change the nomenclature, so this remained top1, but just to add MT to that one to say this is really mitochondrial. This doesn't go to the nucleus. This is for chromosome M, and this is for the rest of the genome, the top1. The relaxation of DNA by topoisomerase at the beginning was discovered very early on when running in agarose gel with ethidium bromide. If you take the DNA you extract, usually it's supercoiled, so it runs fast. And when you put a drop of nuclear extract, you could do it in your lab on this DNA, it'll be miraculous. You'll see all your DNA becomes relaxed. And the enzyme that was doing that then was called the DNA untwisting enzyme, because the DNA is over-twisted here. See the helical structure of DNA, but DNA is over-twisted. And the topoisomerase 1B, without salt, without ATP, and even on ice, will relax this DNA and convert it to a full circle. No break, full circle. And the magic is in this intermediate, which is the cleavage complex, where the topoisomerase surrounds the DNA, cleaves one strand, and then controls the rotation of this broken strand around the intact strand. And we calculated with Keir Newman in this building. The speed is actually quite impressive. It's about 6,000 RPM as these things turn around. And it'll go all the way, DNA realigns, religates, topoisomerase goes, the top1 goes somewhere else. So it is essential, as you would imagine, for transcription and replication. And why? Because when you take a piece of DNA and because of the helical structure, the binding to nuclear matrix, the need to separate the two strands to transcribe and to replicate, and you will understand this if you have a rope. If you take a rope and then Terry takes one end and I have another one, and then one of you is going in the middle and start to separate the strands of the rope on each side, you'll get sort of, overwinding and underwinding on each side. So on one side, you'll get positive supercoiling, and on the other side, negative supercoiling. And if you cannot relieve the positive supercoiling, there is no way you could go back to Terry because it's going to be all knotted, so not knotted, it'll be overwound. So you need something to take away the supercoils. That's what topo1 will do without much effort. It'll take that supercoiled away through these cleavage complexes, And these cleavage complexes is the Achilles heel of the top one. Normally it's very transient, but if it gets stuck, this could be extremely dangerous. And that's what we take advantage of with the anti-cancer drug. And we'll review later that with DNA damage can do that. It could trap topoisomerase 1. And topo1 cleavage complex is also formed during apoptosis, maybe as a way to destroy chromatin. So a few words about supercoiling. This is also in the review. In the context of chromatin where the rotation of DNA is constrained, DNA supercoiling over or under twisting, you can imagine you over twist or you under twist if you go in the sense of the helix, you over twist, you tighten like you'll squeeze the water out of something. And then the other way, you'll just untwist. And the right is whenever you have made so much twist that actually the strand flip on top of the other. That's the right. That's generated. So top 1, M2, MT remove supercoiling by DNA untwisting. They swivel the DNA, whereas the top 2 alpha and beta removes right, meaning they all work at the crossover point, and you will see why later. And there are some basic fights concerning supercoiling. Positive supercoiling tightens the DNA helix, and negative supercoiling opens the helix. And that's very important because for anything to get into the DNA, you first need to open it a little bit, and negative supercoiling is a fact of life. It's actually needed. Nucleosome formation disassembly absorb and releases supercoiling. So when you form a nucleosome, in fact, you constrain some negative supercoiling. When you take a nucleosome away, then you get negative supercoils. And the polymerase generate positive supercoiling ahead. It's logical. They tend to accumulate the supercoils, and negative supercoiling behind. If you have too much positive supercoiling, that will arrest your tracking enzyme and suppress the transcription, destabilize nucleosomes. On the other hand, negative supercoiling facilitate the melting of the strands, and that's necessary to initiate replication and transcription, form D loops, form homologous recombination, and form nucleosomes. But if you have too much negative supercoiling, the DNA can flip, and it can make an R loop, it can make a guanosine quadruplex, it can make zDNA, it can make plectronemes, and that add up very strange structures that are not always desirable. So it needs to be regulated. So these are the two topo 1s, as I mentioned before. The main difference, you see, they're very similar in their biochemistry. The only difference is in the N-terminus. The top 1 MT has a very short mitochondrial targeting sequence, and top 1 in the nucleus has a nuclear localization signal and a long thing that probably talks to other proteins, because topoisomerase 1 needs to be driven to where it has to work. It can't work everywhere. It would be extremely damaging if it were let free. And these are the primitive in some viruses. You get topo 1b, such as the vaccinia virus has its own topo 1b. So the topo 1s, as I mentioned, are selectively targeted by a class of drug which is routinely used in cancer treatment. So topotecan, which is used for ovarian cancer, which is used for small cell lung cancer in second line, which is also used in some pediatric cancers. And then irinotecan, which is broadly used for colon cancer and in Asia and in Japan, in other indications such as lung cancers as well. So they are, both of them in pediatric cancers. And in Asia, there is also this drug belotecan. And this is a natural product, which is derived from a tree, Camptotheca cuminata, which was discovered at the NCI, by the NCI, and then the drug company just evolved these water-soluble derivatives. Today, so these drugs have been approved for about 10 years now, so they are not moving so much. They are still used, very useful. There are new topo 1 inhibitors that are being developed. 3 of them are in clinical trial here at the NCI. We made those drugs, and they are referred to as LMP744, LMP776, andimitecan, and LMP400, andotecan. Andotecan just finished phase 1. We had activity in 1 patient, and the dose-limiting toxicity was bone marrow. Nobody died. The question is phase 2 or no phase 2. 766 is finishing phase 1 now, and 744, we believe, is about to start phase 1 here. So these drugs, the way they work is quite remarkable. So this is the structure of camptothecin. This is the topo-1 nicking closing reaction. So in the topo-1 nicking closing reaction, as mentioned, topo-1 first cleaves the DNA by making a covalent linkage to the 3' end. Normally, the DNA realigns, and this reverses very readily. But when camptothecin comes in the system, what happens, it intercalates in the break site. And not any break site, and that's how we discovered this, hypothesized this mechanism. They were the break sites that tended to have a thymine at -1 and a guanine at +1. So a subset of the topo-1 cleavage sites are trapped by camptothecin as the drug binds in here. So we proposed this in 1990 something, and it took about 10 years to really confirm this. This is the crystal structure. So in the crystal structure, the DNA is green, the break is here, the drug is there, and you can see it's exactly like here. So what happens, the drug prevents the religation, the ligation, and therefore the topoisomerase is trapped. It's not covalent, but it's trapped for long enough that the topo can't get away, and then that leads to collision with polymerases and disaster, and therefore cancer cells die. The camptothecin, because it's a natural product, it was for a long time mysterious why the plants which produce camptothecin, such as the tree Camptotheca acuminata or the trees, why are they not killed by the toxin that they make? And initially we thought it's because the toxin was only made in the bark, but that's incorrect. It's also in the leaves. And what was found is that the camptothecin producing plants encode a mutation on their topoisomerase 1 gene, a single point mutation, asparagine 7222 serine. And this was reported in 2008 by Japanese group. And what was remarkable is the Japanese group had found in the plant a mutation that we had generated or selected in a human leukemia cell line, very resistant to camptothecin, and we had published some years earlier. And that tells you, that's what you define by targeted therapy, is that if you mutate the target as one residue, it becomes immune to the drug. Therefore, the camptothecins are absolutely targeted. And that's even more clear in yeast, because yeast, you can delete topo-1 and yeast can survive. But these yeast deleted for topo-1 are totally immune to camptothecin. And topo-1 is the only target of camptothecin. That's pretty clear. At this stage, nobody has found that it could target anything. And the selectivity is driven by the fact that it just binds into that pocket here, into this cleavage complex of the topoisomerase 1. And that resistance residue is right there, right next where the drug binds, and interferes with the drug binding and renders the enzyme efficient but drug resistant. So the development of new topo-1 inhibitor is going. There was a time where everything slowed down because the main focus of drug development was on so-called targeted therapy, which were the kinase inhibitors, basically. And now it's passé a little bit, so the drug companies are moving into epigenetics, I think, and a lot in immune regulation. But I think there's a coming back of the DNA damaging agents. And we had known for a long time that the camptothecin had limitations. You could cure a mouse. There was a paper in Science in the old days showing you can really cure a mouse with camptothecin, but you cannot cure a human because you can never give enough drug. You are limited by bone marrow and intestinal toxicity. You're limited because the camptothecin, as many natural products, are drug efflux substrate. And you're limited because the camptothecin are chemically unstable. And the chemical instability is due to the fact that they have an alpha-hydroxylactone. And as soon as camptothecin, topotecan, irinotecan goes into the blood, it immediately converts into the carboxylate, which binds to serum albumin. And the only active form is this form. So this is a major liability when you do clinical use of these agents. So the other reason the camptothecins have limitation, they bind very transiently to the topo-1 cleavage complex. So if you wash away the drug from your tissue culture, if you do a tissue culture experiment, it's wonderful because the cleavage complex is reversed immediately. But in a patient, it's not great because you don't want the drug to go away so quickly. So that's why these drugs were developed, the one I mentioned before. So these are the adenoisoquinoline that we developed at the NCI and which are now in phase 1 clinical trial. They are highly potent. So we developed them with purified enzyme and all the tests. They are highly specific for topo-1 cleavage complexes. They are chemically stable because, as you could see from the structure, this is the alpha-hydroxylactone. They have no alpha-hydroxylactone. They overcome resistance to drug efflux, so this is clear. And they trap topo-1 at different sites, and they are more stable. So that's why I think the clinical trials are going on. There is another change in the paradigm of classical chemotherapy today, is that once you have found a warhead, such as a camptothecin or a tubulin inhibitor, what now we could really start thinking and doing is to do targeted delivery. And I'm a firm believer that this is a nice way to go. You could take a very toxic drug, but not lethal, and make sure you concentrate in the tumor. And the camptothecin have been worked on by drug company. You could see here 1, 2, 3, 4, 5 examples of ongoing development of camptothecin warhead associated either to a liposome, to PEG, or to antibodies to target these drugs selectively to the tumor. And maybe then you could do what we saw in mice. If you could increase 10 times the amount of camptothecin in the tumor, you're probably going to get the cures that we saw in the mice. So I think a lot of clinical trials need to be done to look at these derivatives because they may be very hopeful. And one was just FDA approved for pancreatic cancer, Onivide. So let's move to the topo-2. So there are two topo-2s. There is topo-2 alpha, and maybe I think it's better now to call them top-2A, but time will tell. Or topo-2 beta, top-2B. The difference, this is very schematic, but it's easy for you to remember, is that top-2A is replication enzyme, top-2B is transcription enzyme. One reason to know this or to think that way is that top-2A is highly expressed in replicating and cancer cells, and it's not expressed in non-replicating cells. So if you take your heart or your brain, there is no topo-2A, but there is a lot of top-2B. So top-2A is driving replication, and you'll see how, and top-2B is the housekeeping transcription-associated enzyme, and it is expressed both in replicating and differentiated cells. So in neurons, in muscles, and this, you have top-2B, and it goes both to the nucleus and to the mitochondria. So the two polypeptides, so they are shown here. This is human top-2A and top-2B, and with their domain. The cleavage religation domain is here. They're very similar, you could see. 81% identity over that long stretch. And the main difference between the two enzymes is in the C-terminus and in the N-terminus. And the association with other protein will be driven by that. But the active center for topoisomerase activity is highly conserved. It is actually even highly conserved with the gyrase and topo4, except gyrase is made of 2 pieces to make the subunit. So that's why it's a tetramer, but it comes out of 2 instead of 1. In vertebrates, in human, it's 1 piece. In E. coli, it's 2 pieces, but it's highly conserved. See, in this region, the nicking closing activity. And these topo2 work with magnesium, very sophisticated 2-metal binding with a bunch of residue that are drawn here. And this is very conserved when you go from human to E. coli. So the mechanics of these proteins have been conserved through evolution, because that's the way they work, and they work well that way. So what do they do? How do they do the trick? So one way to see the trick is like this. You take two duplexes of DNA. So one is what we call the gate, and the other one is the T for transport. So one will open as a gate, and the other will go through. And you have to think of the NIH gates, because it works the same. It's a two-door. You can't go one door. So first door, the first gate, is you're coming to the entrance, and this is closed. You get inside, the first door opens, and you get in. Then you're in the middle, and then you can go through this opening. Once you're in it, it closes behind you, and then opens on the other side. And then it closes back behind you, and you've gone. Now you're inside the NIH. So this is a two-gate mechanism, and it burns ATP as it does that. So it uses ATP, and it uses magnesium. But it's very powerful, because with this machine, you could take two circles, you could decatenate two circles, you could catenate back. If you have a knot on DNA, it can unknot it, because it will pass a strand through the other. It could knot it, and it could relax, because you could imagine that obviously it can just relax supercoiling. And it will do that pretty well ahead of nucleosome. And it will relax positive and negative. And the only enzyme that generates negative supercoiling is gyrase. It's the only enzyme. The other topo, in general, only relax the supercoiling, positive or negative, but do not generate negative supercoiling. Gyrase does that. So how do they function? How do they integrate their function with the biology of the cell? So let's assume you have a transcription unit, which is with two topological barriers. So you have a piece of DNA, and then there is a promoter here. And the polymerase is there, and you have the RNA here. So as the DNA opens, as we said, you have to generate first negative supercoiling here. Negative supercoiling will enable the initiation of the polymerase. The RNA will get going, the polymerase will move, but as the polymerase moves, and I'm going towards Terry, supercoiling accumulates. Topo 1 will take it away in regions where there is no nucleosomes, just using the swiveling of the strand. Topo 2, if it's just ahead of nucleosome where you have crossovers, will just do its trick and relax supercoiling. So both Topo 2 and Topo 1 will take away the positive supercoils that are ahead of the fork. Now let's look at the replication bubble itself. You have generated negative supercoiling, which is good, but too much, as we said, is no good, because too much negative supercoiling will generate R loops. And R loops will then stall the transcription. You need to take away some negative supercoiling, and this will be done by Topo 1, because you don't have nucleosome here, and Topo 3 beta. And we'll come back to that. Now, in the promoter region, what seems to be happening now is the promoters, to get activated, recruit Topo 2 beta. And Topo 2 beta in the promoter appears to make transient breaks, actually not so transient, that enable the initiation of transcription. And that's where Topo 2 beta is coming back into focus for neurons and for other syndromes. Now, ahead of all this, you have the enhancer. And the enhancer region there are sites of Topo 1, which is important also in the enhancer, not only to relax the supercoiling and to enable the loops of chromatin to adjust, but also probably to activate some breaks and recruit some proteins. And then the Topo 2 alpha and beta could be there. In a neurons, or again a muscle cell, you take away the 2 alpha. Only Topo 2 beta will be sufficient and will do it. If it's a cancer cell, it'll use both Topo 2 alpha and Topo 2 beta to do the trick. Now, if you think of replication and the roles that are very specific of Topo 2 versus Topo 1, so if you take the replication, you first start with the initiation. Again, the initiation of replication necessitates the melting to initiate these fragments of DNA. And when you do that, you'll generate negative supercoiling. But you need that negative supercoiling. Therefore, if you had too much Topo 1, you could not initiate replication. As you initiate this, you generate supercoiling in the flank, and you have to dissipate the supercoiling for the forks to move. So after initiation, when the helicase is moving along, when you generate the supercoiling, the positive supercoiling in front of the replication fork, as for transcription, topo1 will take away the supercoiling in nucleosome-free region, and topo2 will take away, an alpha in that case mostly, in the crossover around the nucleosome very efficiently. So behind the replication fork, you will generate negative supercoiling that could be dissipated by topo1, top2 alpha, and top3 alpha, and then you could form precatenates. When the forks converge, you have a massive excess of positive supercoiling as the forks converge, and if you would not take away the supercoiling, you could never go all the way. So the topoisomerases will take away the supercoiling, topo1 nucleosome-free region, topo2 in nucleosomal region, and when all is finished and everything has been replicated, you end up with a catenane. And that's where topo2, by cleaving 2 strands at a time, will then decatenate. And in yeast, if you take a temperature-sensitive mutant, topo2, it will die because it cannot decatenate the chromosome. Topo3 alpha can decatenate if you have single-stranded region. So I'll come back to that later. The drugs for topo2, there are lots of them. There is etoposide, vepezid. There is doxorubicin and daunorubicin. And then you have mitoxantrone, amsacrin. And what you could see, all these drugs look similar and different. In common, they have this aromatic polycyclic thing and sort of a side chain, but they really look different. And now if you take the antibiotics that are very specific to gyrase and topo4, but not to the human enzyme, although I said they're very similar, you could see they look different. And these are all the quinolones that you probably have encountered: ciprofloxacin, gemofloxacin, moxifloxacin, norfloxacin, drugs that you have heard of before. These are specific for the bacterial enzymes. They don't work on the host enzyme, and those are specific for cancers and for immune disorders, but not against the bacteria. What's in common? Well, what's in common is actually interesting. If you take, for example, here, etoposide, what is clear is the drug works exactly like the topo1 inhibitor. The structure of the topoisomerase 2 cleavage complex trapped by etoposide revealed that the drug binds in the cleavage site, like the camptothecin was binding in the cleavage site of the topo1 cleavage complex, and you get one drug in each of the 2 cleavage sites. You could see it is a very, very intricate geometry, and the drug, the planar ring here, tends to intercalate between the base pairs, and the side chain tend to reach towards specific amino acid that confer the selectivity. So they are highly specific for topo2 alpha and beta. They don't touch gyrase, they don't touch topo1, and it's because they have all these very intricate interactions. And the antibacterial is exactly the same principle. If you take here is the structure of topo4, which is again a tetramer, but see how similar it looks. This is topo2 beta, this is a dimer, this is the topo4, very similar machine, and the drug again bound in the cleavage site with this planar thing stacking against the base, and then hydrogen bonds to specific residues to the bacterial topoisomerase. And that led to one concept, which is that these drugs made us realize that nature, because all these are natural products, developed a paradigm to poison these DNA enzymes, which is to bind the drugs at the interface of the enzyme and the DNA. So that came out, and this is what we refer to as the interfacial inhibition principle, which is very powerful because you could target macromolecular complexes. You don't use pockets in a single protein, you use the interfaces as these things move around, and then you jam them. So that's what, so if you look at them in comparison, for example, in the topo1, you have a single strand break, you stack against the break with the planar moiety, and then you have hydrogen bond to the protein. And for the topo2, you stack and you have different hydrogen bond specific for the particular enzyme. So it's a highly selective process. These drugs do not work at picomolar, so which is another notion that we tend to think that the best drugs are drugs that work at pico-nanomolar. These drugs work, they're not very high affinity, but they're highly specific. They work at micromolar. So nature doesn't need to really get picomolar when it has such a selectivity. All right, we're moving along. So the topo 3. I'll be more brief on the topo 3. So there are two topo 3s, topo 3 alpha, top 3A, and topo 3 beta, top 3B. And I think now we could pretty much be confident over the last 4 years that there is a clear division of labor. Top 3A is linked with replication. It's a DNA topoisomerase. It will work on single-stranded region. So if the DNA is melted, it will work there, but it needs to have a duplex DNA to engage. It will not engage on just single-stranded DNA. It resolves hemicatenane. By hemicatenane, I mean something where only one, so if I could split my arm, only halfway is gone through, not my whole thing. And then you could just cut one strand and it will resolve it. So it's an hemicatenane. And it prevents recombination, because at the end of replication, there are hemicatenanes. Top 3 beta is really the hottest now, is the one where I think we learned the most. It's highly involved in transcription. I think it's a DNA topoisomerase that's coming to resolving R loops. And it's also an RNA topoisomerase. And that's the newer part that we did not anticipate. Even last year, I didn't talk too much about that. So how does it work as a DNA hemicatenate topoisomerase? So if you take a replicating circle, when you finish replication, you have this catenated region, which can be resolved if you have topo 2 by just cutting across 2 strands at a time, and you'll end up with the product. But if you don't do this perfectly, you will end up with some single-stranded region that are hemicatenanes. And these are highly recombinogenic, because if topo 2 doesn't do the job fully, you'll need to do by topo 3. And in that case, it's topo 3A, and it works with Bloom syndrome protein, or with the case with Werner, with the RECQ helicase. So it's a tandem. The top, it's a 1A, which is top 3A. It works with the RECQ helicase. And then the newcomer is the 3 beta. And that came now about 2 years ago, 3 years ago, where it was found. So for a while, nobody talked too much about 3 beta. But the 2 papers came out in Nature Neuroscience. And this one was really what lifted the whole thing, because this was from Finland. And in Finland, there is a region of people who are inbred who have a very high frequency of schizophrenia and mental retardation. So not a region to be at, but that's a lot of these people there. And when they looked at their genome and they looked for what was wrong with these families, they found that there was a deletion in the topo 3 beta locus. And that was a total surprise. Why would topo 3 beta be so important for that? And then at the same time, Wai-Dong Wang in Baltimore at the Institute of Aging, working on topo 3 beta, formed that it was an RNA topoisomerase. So the idea is that RNA is susceptible to make knots. It's susceptible to be entangled, especially during translation. And topo 3 beta is in the cytoplasm. And topo 3 beta is essential to resolve RNA knots or RNA topological problems. And in neurons where you have very long genes, that tends to happen. Or if you have R loops, topo 3 beta will tend to dissolve them and therefore facilitate transcription. But if you don't have the right transcription in a neuron, that shows immediately because of our high level of sophistication. So you end up being schizophrenic or mental retarded if some of your neurons don't function right. So the division of labor between the two topo 3s is they are driven to their site of action by a driver. In the case of topo 3 alpha, it's RMI1, which was also discovered by Wai-Dong Wang in Baltimore. And RMI1, together with Bloom, engages topo 3 alpha in replication intermediates and resolve the hemicatenanes and holiday junctions, and therefore it's critical. In transcription, the partner is TDRD3, which which is an RNA binding protein, and TDRD3 then engages topo3 beta on the RNA and on R loops with the fragile X complex as well. So this is emerging as being very important, and that topo3 beta is much more important than we had anticipated before. And that leads me to what I want to finish with, is the role of topoisomerases in genomic integrity and human diseases, which is something that is really emerging now. So not only drugs, but DNA alteration and physiological processes lead to the formation of persistent topoisomerase cleavage complexes. And at least for the one enzyme, TOP1 and TOP2, we have many examples where the persistence of cleavage complexes lead to genomic damage. So I'm not going to say anything about the anti-cancer drug because we have covered that and we use it therapeutically. But if the DNA has oxidative DNA lesion, very common, 8-oxoguanine, that poisons topo1, that poisons topo2, both in the nucleus and in the mitochondria. If you have AB6 sites, same thing. Topo1 gets sluggish, it's going to get caught. Carcinogenic base adducts, so tobacco products such as benzpyrene, induce and trap topoisomerase cleavage complexes. Both topo1 and topo2, NICs, UV lesion trap topoisomerases. Ribonucleotides, I'll come back, and I'll show you some examples. So why would that be so bad? Let's say you have a topo1 cleavage complex, which normally is very reversible and needs to go on and off and very quickly. So normally it's coupled with replication, and replication follows. But if this is stuck, the replication can collide, and that will generate what we call replication runoff. It just collided. And now topo1 cannot religate anymore. This is irreversible. And that leads to genomic damage. And I'll tell you at the very end how this is resolved. Or another trick is that the fork collides, but it can reverse. So it's what we call the chicken foot. And PARP is involved in this reversal to some extent. But it is a problem. So that's one example of a problem. I'll give you some other mechanics, and they are in the handout. We could take one. Take this one. This is a topo1 cleavage site, which are very frequent. They are in the genome. There are many, many if the DNA is not nucleosomal bound. Let's assume you have a NIC. Topo1 is oblivious to the presence of a NIC. It will still cleave here. But if you have nicked here, and you have topo1 making here, then this makes a double-strand break. And the double-strand break, then the DNA comes apart. So what you've done is a single topoisomerase molecule has converted a NIC into a double-strand break. And other things like this, if you have, for example, a NIC on the same strand, topo1 cleaves upstream, now you have that little piece here, and that can fly off because it's not held by topo1. Now this could religate across, but it will form a bulge. It will form like a slippage. So you'll get genomic instability. And all these are described in the review. So if you're interested in topoisomerase gymnastics, you could look there. And the most recent, which is still a very ongoing work, both in our lab and many labs now, is the interest that we think of the genome being all DNA, but what's coming up is there is a lot of RNA that gets incorporated in the genome on purpose or by accident. And the ribonucleotide incorporation in DNA by DNA polymerase is actually very frequent. It's been estimated in yeast, for instance, that it could be 1 in 1,000. And in yeast, normally, this newly incorporated ribonucleotide is removed by RNase H2. And RNase H2 is essential. So if you have a mutation in RNase H2, you'll get a lot of these ribomis incorporated. Well, how bad is this? Well, it's pretty bad because you have a ribo, eventually the polymerase gets stuck, but what's even worse is if topo1 comes and here before RNase H2, it will immediately convert this into a NIC. And the way it does it is topo1 binds to the end here of the DNA on the 3' end, and because it's a ribose and it has a 2' hydroxyl, the 2' hydroxyl then attacks the phosphate again, phosphodiester here, and then liberates the topo1, but generates a 2', 3' cyclic with a nick. So what topo1 does, then it acts as a ribonuclease generating a nick. And that could be pretty bad, and it's been shown in yeast now, that if that is generated, a second topo1 site could be formed right upstream. So that's what I showed you right before. And when this forms, this could then dissociate, and topo1 will religate across, and this will form two base deletion. And in repeated sequences, you will end up with this deletion. Could be repaired, but that's a problem. The other one, which I already alluded to, and work that we're finishing now, is that if you have made a nick on one strand and release topo, topo could just jump across and go to the opposite strand. And if it finds a site right there, it will convert this into a double strand break. So a single ribo in DNA can generate a double strand break just with topo1. And we could do that back in biochemically. So what are the human diseases linked with topoisomerases so far? And I think the list might be expanding. In the case of topo1, we're looking at neurological diseases due to the lack of removal of topo1 cleavage complexes. And I'll give you a little key at the end of why this is. Topo2b, when topo2 cleaves the DNA, it needs to religate it, especially in the promoters. If it doesn't religate quickly, another piece of DNA can come and do a translocation. And the causal implication of topo2 beta is now pretty well established as a source of leukemia, secondary leukemia, but even primary leukemias, and in translocation site in prostate cancers, when the androgen responsive genes, there is a topo2 beta site in the promoter, and that could lead then to recombination. In the case of 3 beta, as I indicated earlier, the lack of 3 beta leads to neurodevelopmental disorders, schizophrenia and cognitive impairment. And then, if you have a lack of repair of these topo cleavage complexes, you end up with this type of disease. Neurological disease in the case of that enzyme, tyrosine DNA phosphodiesterase 1, SCAN, spinocerebellar ataxia and peripheral neuropathy. And in the case of TDP2, intellectual disability, seizures, and ataxia again. If you cannot remove the topo cleavage complexes, which is what these two enzymes do, and I'll show you how they do it. So the way the topoisomerase cleavage complexes are repaired, and because of the danger, all species going all the way to yeast have a battery of enzymes to remove the topoisomerase cleavage complexes. And when these enzymes are deficient, that's where you end up with these diseases. So how do they do it? So you could look at the review where you'll find a little scheme the same. So if you have a topoisomerase 1 cleavage complexes, so topo 1 is linked to the 3' end of DNA. You could see it here. So this is the DNA. Okay, it's linked to the 3' end. There is a specific enzyme which was discovered here by Howard Nash, which he called tyrosyl DNA phosphodiesterase. Tyrosyl stands for the fact that this is a tyrosine linked to the 3' end. DNA phosphodiesterase because the enzyme will cleave in the middle here. And what TDP1 does, it removes the topo 1 at the site of linkage and regenerates a 3' phosphate. So that's TDP1. And if TDP1 is deficient, topo 1 cleavage complexes accumulate, and then you lead to this neurological syndrome. In the case of topo 2, there is a specific enzyme, at least in human invertebrates. In yeast, it's the same enzyme. TDP1 does both. But in humans, you get a TDP2 enzyme, special. And you could see the polarity for TDP2 is that the topo 2 is linked to the 5' end. So TDP2 will again, tyrosyl DNA phosphodiesterase 2, removes this topoisomerase 2 and regenerate a 5' phosphate. If you don't have TDP2, that accumulates probably topo 2 beta cleavage complexes in neurons, and that leads to intellectual disability, seizure, and ataxia. But because it's so important, cells do not rely just on the TDPs. They have a backup and alternative pathway, which is to chop the piece of DNA a little bit upstream of the topoisomerase linkage by nucleases. And these nucleases are very commonly known, like MR11 nucleases, CTIP, that could cleave these, and then that will require some gap repair. So all our work is on the topo-1 inhibitor is with the collaborator at Purdue University. That's why we could put this drug in the clinic. The NCI director for translation research, James Dorosho, we're doing the clinical trials with him. And then the developmental therapeutics program here, we're very connected to them. So if you have any question, I'll be happy to take them.
How common are these neurological disorders caused by the deletion of the topo-3 beta? How many people are involved?
Well, it's hard to tell yet. And it's hard to tell whether the heterozygotes have minor forms of that. And
Very new, I guess.
it's very new. I think it's very new, and also very new that the TDP2 patients, those patients, it's also very new. I didn't put the date here, but this was found in a patient who had these intellectual disability seizures and was found that was homing on TDP2. So we're just starting to scratch the tip of the iceberg, I think. Okay, you're all burned out?
It's late.
Yeah, okay. You could look at the slides. You could send me an email if you have any further question, or come visit me.